So today we're going to have our second lecture on the biogeography of the ocean. And last time we spent some time talking about what biogeography is. And of course, we very quickly settled on the definition that it's really the study of the distribution patterns of organisms on Earth. And part of understanding what the basic unit of biogeography is was the species. And we spent some time last time talking about what a species was, how it's an esoteric question, but we discussed a little bit in class the arguments from Jody Hay that it's really difficult to come up with a single definition of a species, and that's led some people to despair about the science of biogeography. But from a practical standpoint, in the ocean, what a species is really just a morpheme. And what that is sort of an organism that has a significantly different shape that we just assume that it's a species. And of course, Charles Darwin said, that there's many definitions of species, but every naturalist knows vaguely what he means when he speaks of a species. And so what Darwin is getting at here is that the whole idea of species is what's called a tacit recognition, or it's tacit knowledge. It's something that you recognize, but there's more than what you can say about it. And so we last time developed this idea called the biogeographic hypothesis. And the biogeographic hypothesis is that there is a strong link between the physical environment and organisms. In fact, it's strong enough to say something about either of these given the attributes of another. So for example, you may or we may understand the physiological tolerances or temperature tolerances of a certain fish in the ocean. And we understand that association not only physiologically, but where it's found in the ocean. And we can make projections into the future of what future temperatures might be like. And because temperatures are going to change, we may say that we think this species is going to decline. That's using the biogeographic hypothesis. We understand what that link is, what that association is now today, both physiologically and in the environment that we find, and we make projections about what we think. We make projections about the future environment and what we think the species might do in the future. We could also use it in the reverse to go backwards. So let's say that we are digging for fossils and we find fern leaves in a desert somewhere, fossilized fern leaves. Well, we would deduce that at some point, based on the organisms that we've found here, that these ferns may have inhabited, a swampy or a moist situation, certainly not the desert that you found them in. And so that's a way of looking at species distributions and saying something about the environment. So we can use the biogeographic hypothesis in either way. Now, in the study of biogeography, there's another unit that we should talk about, which is a biome. Now, the general response to what a biome is that it's a distinct group of organisms living together with a certain physical climate. And in fact, some people say, why even bother with this idea? And the reason why is that an understanding of biomes allows to control for variance in biological responses in different environments. And so what I mean by that is that if we went to Delaware and we found that there were no baboons, does that mean that the species baboon is not doing well? And the answer is no, it doesn't mean that at all, because baboons are not found in the biomes that are found in Delaware. They're not associated with each other. So not finding them is not a big problem. Now, if you go to where baboons are found in Africa, and you know what that biome association is, you could track their populations either up or down and say something about them sensibly. But underlying those differences is really understanding the nature of biomes, right? So that's what I mean, is that it helps to control for variance in biological responses in different environments. So you may go out looking for a certain biological response, but if you're in the wrong place where you're not expecting that response, then you are you may be out of bounds. And so that's why this idea of a biome, of a distinct group of organisms living together within a certain physical climate, maybe it seems naive, But it actually does underlie quite a few of our analyses of trends in organisms over time. And so it's important. Now, typically, when you ask what a biome is, you're going to get some kind of undergraduate map that looks something like this. That we've got tundra, boreal forest, temperate forest, and then all sort of like states that are carved out on these maps like this. And they're more or less like drawings, and the ocean is essentially all the same sort of bio. Now, we can ask, when we look at all these different categories, what are the major sort of descriptors? And you come up generally with four. One is that a biome has sort of a description of a physical climate. Is it hot, cold, windy, ice cover? There is some sort of hard substrate, like is it rocky, is it sandy, is it topsoil? What is the primary producer, right? So what is it? Is it grassland? Is it a forest, right? What is the thing that's fixing CO2 into organic matter? And then finally, the timing, the phenology of the system. Are there strong seasonal patterns? Are there migrations? What's happening there, right? And so those are general characteristics of biomes when we get to the terrestrial system. Oh, what about the ocean system? Well, we could go into situations, say like coral reefs, and we could describe a physical climate. It's clear water. Right, so coral reefs are generally found here in the tropics. There's no turbidity or sedimentation. There's generally high salinity, bounded by 20-degree isotherm, exclusively tropical, right? So the substrate is rock or carbonate. Primary production is symbiosis between coral and zooxanthellae, and we'll talk about that later in another lecture. And there's not much seasonality, but there's these large sort of, quote, hatch events, right? Well, what about one that's a little closer to home in Delaware, the salt marsh? Clearly, maybe that's some sort of biome or ecosystem that we can recognize, right? The physical climate is the intertidal. There's high turbidity and sedimentation. The substrate's mud, sand, maybe some rock. The primary producers are seagrass and halophytes, which are salt-loving plants. And, there's typical greening and flowering in the in the seasons. Another one that I find fascinating, one that I spend quite a bit of my time in my youth, were things like kelp forests. So the physical climate was sort of clear, nutrient-rich, cold water, preferably turbulent. There's a hard substratum or rock on the ground. The primary producers are giant kelps. These are super productive, right? So they grow this Macrocystis porifera, which we see here, grow to 50 meters tall, growing up to 60 centimeters a day. Huge amount of primary production. And of course, there's general phenology and seasonality with that. We can even go to the deep sea, right? There's a physical climate. There's high temperature, high pressure, there's no light, the substrate is rocks. In fact, we'll hear more about these in a different lecture. The primary production is more or less chemosynthesis, and there's really not a whole lot of seasonality, but there's some explosions. And so the question here is that, well, yeah, right, so is there anything really wrong with the biome concept in the ocean? In other words, when we get to the systems that we're talking about in the ocean, right? These ones that we touched on here, the physical climate's very cold, the very hot, the dark to light, turbid to clear, rocks to mud. Is it just the case that biomes in the ocean are just sort of they're just like terrestrial biomes, except we've thrown water on top of them? It's just a flooded landscape. That's all it is. And so we add some water on top, and that's all there is really to think about. Well, there's a question. What do these things really have in common when we think about these? When we think about coral, we think about seagrasses, or salt marsh, we think about kelps, and we think about hydrothermal vents. And the big thing is that we have ocean plus bottom. We still have a solid substrate that we're attaching to. And that is a key feature, because actually most of the ocean, remember its volume, is not like that. Most of the ocean is open water with no solid thing to attach to. And so the physical climate, the substrate is water, and the primary producers are photosynthesis by phytoplankton. And so what when we ask again what the basic physical climate of the ocean is, we get back to this graph that we had in the very beginning of the class, right? So we're talking about an intersection between light and nutrients. And if you can solve that problem between light and nutrients, then that is the physical structure that actually makes an ecosystem go around, right? And what are the physical mechanisms that set this up? Well, things like temperature, freshwater input, wind, the degree of mixing, mixed layer depth, nutrients, light, particles in the water, all these different things. And in fact, one way to think about this, and in fact, the ways that people have gone about trying to understand biomes, not in terms of things that are attached to the ocean floor, like coral reefs, but things that are the majority of the situation in the ocean, which is up in the water column, they do it based on trying to understand this dynamic. And the thing that changes this dynamic generally is the degree of mixing driven by wind. And so we generally think of basically four ocean biomes that correspond to zonal wind patterns. And so here's the zonal winds. We have prevailing easterlies, prevailing westerlies, tropical easterlies, these are the trade winds, and then we reverse the pattern in the southern hemisphere. Now, in order to generate these wind patterns, basically it is driven by the latitudinal imbalance in Earth's radiation. So when we take a cross section here from 90 north to 90 south, we generally have net heat gain here in the tropics, and we have net heat loss out into space. And so there's a net energy transport away from the equator to the North Pole. So generally, heat is transferred from the equatorial regions toward the poles. And there's two ways to do this. There's the latent heat of water through the atmosphere and ocean currents. There's two ways to do that. So really, when we're looking at the circulation patterns of the ocean or also the atmosphere, we're really figuring out how to actually take heat and move it towards the poles, because we actually have a heating imbalance. We have more heat coming in here at the equator than we do at the pole, and so there's a net energy transport towards the poles. Now, when that happens, when we have, what do winds look like? Well, let's think about this for a minute. So we're heating the poles. That creates rising air, or low pressure in this case, and that is then replaced on the surface by winds coming in from the poles, right? So you can think of it this way, right? So we have rising heat. That heat actually gets transported up into to these polar regions. We actually have a collision, right? So we actually have a higher pressure here, and then because we have high pressure in the poles, we have high pressure moving back towards the low pressure. And so generally, on a non-rotating Earth with no continents or Coriolis effect, the wind pattern would be very simple. The air rises at the equator, sinks at the poles, and the heat is transported from the equator to the poles, and the winds generally blow from the poles toward the lower latitudes. Now, when you start to spin the world, you generate the Coriolis effect. Now, we're not going to go into the Coriolis force here. That's something that you'll get in physical oceanography. But generally, what tends to happen is that as these winds, these were surface winds coming toward the pole, they tend to bend. In fact, in the northern hemisphere, they turn to the right, and the southern hemisphere, they tend to turn to the left. And so you tend to get these bands of zonal winds. These are blowing, in this direction towards the west. In fact, they're called easterlies because they're coming from the east. They're blowing towards the west. And here's the westerlies, now blowing from the west to the east. In fact, so here's surface winds. They tend to turn to the right. And so we're not going to get into that, but these are basically how the four zonal wind bands are formed. Now, because of that, there are basically four ocean biomes determined by wind forcing. And so we can talk about these descriptively. So generally the polar system, right, this is sort of north of 60 degrees. There's winter ice cover and moderate wind speeds at high latitudes that constrain mixing, but usually get strong enough to mix nutrients to the surface. So here's an image of seasonal sea ice here in Antarctica. Again, here's ice, here's icebergs. There's plenty of ice in these cases. There's also plenty of wind, lots of mixing. We generally have very limited light source. So here's a light curve basically from McMurdo Station. You can see that these are days, and this is basically the amount of sun that's coming up. You can see that in June and July, wintertime down there, there's no light. So really this is a light limited system, right? So this is a production is highest when you have the highest light. Compare that to, say, a trace off of San Diego, California, where you have light pretty much year round. And so because of that, you have a trace of nutrients, plant, biomass, and water temperature that goes something like this. Water temperature is fairly stable because it's a polar region. It's cool, generally cool there. Pretty stable during the year. Nutrients are generally stable, and the reason why is that it's well mixed from the winds. And plant biomass is light limited, and so you really get big blooms during the summer, and they turn off in the fall, and really nothing in the winter. So these marginal ice zones is where the ice pack meets open water. Areas of shallow brackish surface water due to brine rejection during freezing. So what we're looking at here is this is the formation of sea ice, and so what you can actually see here is that as we cool the surface in this region, salinity is increasing, and as you form ice, you basically are rejecting the salt. That's why the ice floats in that situation. It pushes those. As the ice forms and forms a crystal lattice structure, it rejects the ions that make the salt, basically the sodium chloride, and so you get hypersaline water right underneath this, and that basically sinks down to the towards the bottom, and it sort of creates these interesting mixed layers. We know that these retreating marginal ice zones have enhanced production, so as ice goes away, we generally see increased productivity. That's this graph over here, increased annual productivity, and that's as the ice goes away, you alleviate the light limitation and you get increased production. There's generally high levels of siliceous ooze, and what is that? Well, generally we have high diatoms in these regions. So this is opal in the sediment, and opal is basically siliceous ooze. It's left over from the diatoms, and they have sunk down into this region, and so you generally have high silica or high siliceous ooze in these regions, and this is just sort of a cross section of what these deep sea sediment cores look like. So up here is your chocolate-colored terrigenous red clay, typically structureless with few organic remains and occasional fish teeth. That's in the open ocean. And then here's the bottom diatom ooze from the Antarctic Ocean. It's high concentrates of a diatom, gives sort of this fluffy aspect. That's the ocean. That's the bottom of the ocean there. Also in the polar levels, it's generally characterized by low taxonomic diversity at all trophic levels except for the mammals. In fact, if you take a look at the endotherms, or phylogenetic diversity, they are the highest up here in the polar regions. There's plenty. And compare that to, say, ectotherms and sharks. There's just not a lot of diversity in sharks in these Arctic areas, and that's something that's called metabolic asymmetry. There's an interesting science paper here about that. But basically what it's saying is that when you're cold, you need to, in order to be active and productive as a good hunter, you got to be able to generate your own heat. And so that's why you have sort of marine mammals really dominating these regions. At the next level down, we have prevailing westerlies, the westerlies biome. And in this case, the mixed layer depth is strongly seasonal. The mixed layer deepens with winter, with increased wind stress and convective cooling forced by seasonal changes in sun angle and cloudiness. Shoaling of the pycnocline occurs in the spring with response to development of a warm of a water column stability and increased solar radiation, and deepening of pycnocline occurs in the fall with convective cooling. So this was the basic idea that we were working with in the first part of the class, right? We were really working in the westerlies biome where we have these seasonally dependent mixed layers, seasonally dependent solar forcing, and that actually really drives productivity, right? So there's pre-bloom conditions are high nutrients at the end of strong winter winds due to mixing, and then there's a bloom initiated at the spring when there's plenty of nutrients and light. So we're getting back to that Sverdrup idea. That bloom becomes nutrient limited when nitrate or other nutrients are exhausted and it crashes, and then biological regeneration takes place at a, in fact, lower scale. And so here's what so P here is productivity, right? In the wintertime, we have things that are really well mixed, right? So this is what basically the nutrient concentration looks like. It's well mixed. As we start to get a little more in the early spring, we get some stratification. We have a bloom. We start to draw nutrients down, and then in the summer, nutrients go really low. Our bloom now sort of subsides, and then we have a secondary bloom as we start to mix nutrients back up into the system in the late summer. And so the typical trace here for the westerlies biome is one that we've seen in our course so far, is that we've got varying water temperature. We go from very cold to very warm, back to very cold. And our nutrients start high in our light-limited wintertime. They get drawn down during the spring and the summer. There's another little shot of nutrients that comes up into the fall and then off into the winter again. And our plant biomass, again, our phytoplankton, low in the winter. Spring bloom starts to crash again when nutrients run out in the surface, and then as mixing starts to kick in the fall, again we get another little shot of nutrients, it's another little secondary bloom, and then off we fall. Now the next biome that we're going to talk about is the trades biome. This is synonymous with the tropical ocean and extends from 30 north to 30 south. And this trades biome covers about 45% of the total area of the ocean. It's gigantic. So this is basically the dominant form of the ocean that we have out there. Seasonal radiation flux is such that the mixed layer is maintained continuously. There's no seasonal shoaling from convection, right? So here in the polar system, our temperature profile is basically isothermal. In the temperate region, we have a seasonal thermocline that comes and goes, but in the tropical system, we have a permanent thermocline. It's always warmer up here than it is down here, and so it's hard to always mix nutrients up seasonally. So the levels of solar radiation is such that autotrophs are less commonly light limited. They're more nutrient limited because they're locked. There's really no access to those deep nutrients. And the pycnocline is coincident with the tropical thermocline. It has very high stability. It's very difficult to mix. And so in these trades biomes, there's not many large blooms. There's some. There's large swaths of nitrogen fixers. So if you can't mix nitrate up from underneath, you're going to fix it. You're going to fix it in using nitrogenase. And of course, these areas are impacted by alien dust because we know that nitrogen fixation now requires iron that comes in from these dust areas. Again, here's some examples of trichodesmium lines here. So also, right, if we look at these typical traces, nutrients are generally low year-round, water temperature is generally high, and plant biomass is generally low. Now, one of the components here of the trades biome is that there's actually really large diurnal migrations. So I don't have the scale here on the left here, but these are basically acoustic echograms of migrations of zooplankton coming up and down, right? So they can move from 200 to 500 meters, which is amazing. And the reason you can do that is that you have light that penetrates that deep because the water's so clear. And so the water's so clear because you don't have a whole lot of plant biomass. And so vertical migration is a big deal there. It happens everywhere, but it's especially dominant in this trades biome. Now, the fourth one that we're going to talk about is really this coastal boundary zone. We're not really going to talk about these big zonal winds, but really we're talking about winds along the coast. So this is a topographically diverse and difficult to generalize habitat. It's highly variable. Generally has a boundary with continents, has a large impact from rivers, and coastal upwelling is a major feature. Remember, we spent some time talking about that in the beginning of class. Again, this is a lecture, or this is a slide from a previous lecture. Here's cold water here off of Delaware and New Jersey, and in fact we get phytoplankton blooms as we get upwelling. And so that's just an interesting aside that we think of the coastal biome kind of as its own thing. But having these large biomes is not terribly satisfying for a lot of folks. In fact, some people said that these biomes are just way too big, and if you spend time out in the ocean, you might think that, okay, we get these huge sort of wind-driven biomes, but we know other parts of the ocean that seem to operate in really, really different ways. And this was an idea that came from Longhurst in the late 90s and early 2000s, that he said that, maybe instead of these huge biomes, what you really want to do is think about these smaller subsets that are called provinces. In other words, we know that something like the California upwelling system is fundamentally different than the open ocean, so just putting this all into a trades biome or into a temperate biome doesn't really make sense. We know there's big differences between these systems, and so he set out and made a map that looked like this, sort of these rectilinear lines. And in a lot of ways, I'm really sympathetic to this. We know that different parts of the ocean actually work in really, really different ways. But of course, there has been pushback. People said, is this even really advised, right? It seems weird. Maybe it seems right to draw these state-like boundaries on the land because we know that the substrate's not really moving around. In the ocean, it is moving around. Does this make sense? Like if I drive a ship across this from 60 to 56, is there going to be some kind of boundary there? Is this even really a thing that we should even do? And so there's been some resistance about categorization of the ocean in such a way. But I want to assure you that this is actually really much up and coming, especially when we talk about marine protected areas or exclusive economic zones or territorial seas. There are lines that are being drawn in the ocean to sort of protect a place or to categorize a place, but this is maybe a little bit of a problem because this is a fluid system. We know that there's exchange between these regions, and so does this even make sense? So let's just digress just for a little bit and talk about what it means to categorize something. So let's suppose that we actually have a distribution of things. It could be water temperature, it could be the lengths of fish, it could be whatever. And in this case, I drew 1,000 random numbers that have a mean of 10 and a standard deviation of 2. And in fact, when I compute what the average of this whole population is, it's 10.02327. Now, let me ask you this. This mean value actually is not a value that even occurs within this 1,000. In other words, the mean does not occur in the population. And in fact, rounding to 2 decimal places, 10.02, it occurs only one time. And in fact, rounding to 0 decimal places, it occurs 200 out of 100 times. So there's only 200 quote tens in here out of these 1,000 things. And the big thing to remember here is that means are properties of populations and not individuals. Means are properties of populations. And so this is one of the reasons why stereotypes or classifications can be bad. They don't actually describe any particular individual, right? And so that is the major objection to doing this, is to say, okay, you're going to say that this big chunk of ocean has an average of this property and this property and this property, but when I actually go to that ocean, do I find something that has those properties? And the answer will be most likely not. Most likely not. And the reason why is that these categorizations don't necessarily describe any one part of the ocean there, but it's a property of this gigantic space. And so the question comes down to say, well, why should I make a space that's this big and not one that's a little bit smaller over here and one that's a little bit smaller? What determines the sizes of these things? Well, in other words, does it make any sense to make some sort of categorization like this? Well, let's talk about an alternative to this. What happens if we really narrow in the standard deviation? That here in this case, our mean is 9.99, right? This mean does not occur in the population. Rounding to 2 decimal places, it occurs 42 out of 1,000 times, and rounding to 0 decimal places, it occurs 1,000 out of 1,000 times. So even though these means of these properties may be properties of the individuals, depending on the variance, it actually, this mean describes this distribution fairly well. And so here's just a key here, is that these summary statistics that we're going to apply over these provinces, they're simple models that may or may not apply to any individual of the population. In fact, George Box, we got his little quote here, he says that all models are wrong, but some are useful. And that's what we're getting at here when we're talking about turning these things into provinces, right? So what Longhurst and others have thought is, they said, look, we think that maybe this part of the ocean out here, yes, we understand that just drawing a rectilinear space and saying this is one type of group over here can be problematic, especially when there's lots of distributions across or a broad standard deviation across a category. But if it turns out that it is actually low variance in this region of the ocean, it does make sense to group it together. And so Longhurst basically says that grouping these provinces, the idea is more like this and less like this. They're basically areas that actually, they do cohere together. They actually are similar to each other, even though we can't find the exact, average in there. And this gets down to a basic idea of how it is that we want to actually go out and do science, right? So is it worth making models like this that we know are not perfect descriptors? We know that categories can be problematic that way. Well, you divide up any group. It could be a group of people. It could be a group of animals. It could be groups of the ocean. We know that summary statistics about entire groups maybe are difficult because it's hard to find an individual that satisfies all those conditions. But it could be a useful description of a group in general. It could be. And in fact, that's what Longhurst is getting at here, right? So he thinks, I'm going to go ahead and put this out, and I think that I'm going to draw these lines, even though I know that there's fuzzy edges on either sides of these things. No one's going to believe that these things are exactly categorical like that, but that's okay. They are still useful. And that's worth thinking about, because there are categories. So a lot of people say, we don't like categories because there's so many cases that don't fit into the categories that they're just wrong. They're bad. Well, I don't know. Yes, some of these categories have fuzzy edges. They don't quite match up, but they can be actually quite powerful. And we're going to talk about what that is in a minute. Now, compounding Longhurst's problem is the fact, and he wasn't able to deal with this, is that the ocean is actually, the substrate is actually moving around, right? So these are the major currents in the ocean. That's the substrate. That's actually moving, right? So that's what we're talking about. Is the land moving? Yes, really, really slowly, right? So incredibly slowly. But is the ocean moving around quite frequently? Yes. And so one of the questions is, can we actually dynamically map these water masses or these provinces in space and time? And why would you want to do something like that? Well, right, these water mass boundaries or these provinces are predictive of hydrography, and changes in province size or water mass size and distributions are maybe bellwethers for cyclical and secular Earth cycles. And you can actually do a true comparative analysis by controlling for advection. So let me just show you how you can actually do that. Now, you remember this problem, right? What is a species? We understand that species is hard to define, right? We sort of recognize it when we see it. But the reality is that, you know, when we are studying the tree of life, we use this one in the previous part of the course. You know, this is the eukaryotic tree of life from Sandy Baldouf in 2003. You know, yeah, there is a continuum along, you know, there's an evolutionary continuum along these branches, but there's also these split outs, right? So are these are categorically different than these groups over here, and this is categorically different than this one. And so how do you decide? How do you decide? Well, in this case, what you do is you take a whole bunch of gene sequences and you ask a computer to cluster them. You ask a computer to cluster them. And so what are we going to do in this case? Well, what we're going to do is we're actually going to take the measurements that we have from ocean color, in fact, two radiances. One's blue light coming out of the ocean and one is green light coming out of the ocean, and the other is temperature coming out of the ocean. And we're going to ask a computer to do an unsupervised classification to say, give me the major groups. Give me the major groups. And in fact, you can probably see that, hey, you know something? There's lots of sort of groups in here that are really similar to what Longhurst said, right? So if we basically take observations and we classify them using algorithms that are actually similar to the way species are classified, you can see, yeah, look, here's this chunk over here coming out, you know, and here's, you can see the Gulf of Mexico has got a, you know, and the Caribbean are all grouped together. And then we've got a couple different divisions in here, you know, here's the Gulf Stream. And so what you're looking at in this map, these colors just mean similar types. That's all they are. So this type is similar to this type, similar to this type. So these are similar characteristics coming from remote sensing. And so here's the major question. In fact, the one that we wanted to answer, right? So that we said before is that if I was in 60 over here and I drove my boat over here to 56, if I crossed this line, would I see anything? Well, I don't know. If we ask a computer to make those supervised classifications and then basically just look at the edges, in fact, just look at the boundaries, and that's what we have here. Here's basically the boundaries, right? All these black lines are the boundaries between these. And then here's actually a track of a ship driving out. Here's salinity, right? So take a look at that. Look at that break in salinity from basically 34 to 36 right on that boundary. Now, salinity isn't being detected by these sensors. And so, yeah, it really looks like in some cases that, yeah, there is a hard line in the ocean that you can cross. So here's some examples. Boom, here's a nice hard line here, right? Look at all these different analyses that come up. And in fact, although there's some places it doesn't work, right? So here's the middll of the subtropical gyre, Pacific and the South Pacific. You can see like this, you know, nice gradient. So maybe you don't trust this boundary as much as you would trust this one. So there are hard boundaries in this thing, and it's pretty interesting. And so you can actually, let me just show you what this actually looks like in space. I'm going to load this up. Here is dynamic boundaries now of these provinces in space and time for the MODIS Aqua mission, right? So what you're seeing are these similar colors are actually similar types of water types, and you can actually see them evolve by month or change by month. And so you can, so these provinces are actually moving around. So what have we done? We've made provinces. So what have we done? We've made provinces that kind of look like, Longhurst boundaries, and now we can actually animate them in space. Now, why would you want to do something like this? So here's an idea. We know that one way to detect El Nino and La Nina is by looking at the amount of upwelling and the amount of ocean color in this region here off of South America. And in fact, if we take a look at these ENSO indices. So these are three popular ENSO indices: the Southern Oscillation Index, the ONI, and the MEI. These are all indexes of El Nino. And we compare those to the area of these biomes, over time, you can see that actually they follow the same kind of pattern. In other words, the areas of these things here over time actually do carry with them a climate index, which is pretty cool, because so now we've taken these states that Longhurst maybe was a little bit nervous about, and maybe we were nervous about, and added some dynamism to them, and we found that the areas of these provinces in the equatorial Pacific actually are decent indicators for things like El Nino. Now, what about other things, right? So we can actually look at the areal fraction of globe. In fact, just come down here to the lower left here. This is the areal fraction of the globe that is covered by ocean deserts, those big pink areas that we were talking about before. And one of the things that you can see here is that ocean deserts seem to actually be going up over time. So from 1998 to 2008, it looked like that there is a real increase in ocean desertification, which is pretty cool. And so one of the ideas here is that these pink areas are getting larger. So these areas that are really low in productivity, and they're overtaking sort of these less deserty areas over here. And so it looks like, at least over those years, it looks like that we have the ocean deserts growing. And in fact, growing quite rapidly. In fact, the areal fraction of globe here that they grew over a decade is sort of equivalent to about 50% of the size of the United States. It's pretty good size. And in fact, these areas also seem to be related to the Pacific Decadal Oscillation. So here's the Pacific Decadal Oscillation here. And you can see here our area, this is our total desert province areal fraction, sort of goes up and down with Pacific Decadal Oscillation. So actually it has a climate signal in it. And in fact, the Pacific Decadal Oscillation has been implicated in fish catches all over the world. In fact, even evident in tree rings. So beyond that, these provinces, right? So here's a shot of a transect that we were on a cruise. Here's a shot of the provinces that we went through, and we actually sampled the flavobacteria communities. It turns out that they group together according to province, right? So this is a tree of flavobacteria communities based on their similarity, and you can see that they actually group together all in the same type of province here, right? So here's 6 and 4 together. Here's the green ones. These all group together, right? So it's actually pretty amazing. So and 17, 18, these all group together. They're all in the same province. So that's bacteria, that's climate. What about bigger things? Well, we can actually test this idea on fish too. So we can use these dynamic seascapes, is what we can call them. They're basically dynamic provinces to predict the marine occurrence of an endangered species, Atlantic sturgeon. And so what can we do? Well, let me just tell you a little bit about Atlantic sturgeon. One is that they were a Delaware fishery that collapsed. Like lots of other fisheries in this region, collapsed during the turn of the century, and now we've got some low level of fish here. They can be actually quite large. They're gigantic. This is a picture taken near Barnegat Light, New Jersey, right? So this is like, 7 guys, 1, 2, 3, 4, 5, 6, 7, 8, 9 guys here, quite a few. And these guys are actually taken by commercial fisheries. There's about a 13% mortality each year, and they are in fact now endangered. And so Matt Brees, Dr. Brees now, he wanted to link ocean conditions to Atlantic sturgeon occurrence to try and make predictive models to avoid sturgeon and sort of keep fishing. And so what he wanted to look at was in fact these seascapes. So here are these seascapes, which are basically the same thing as these dynamic provinces. It's the same global algorithm. And these black dots here are actually listening stations for tagged Atlantic sturgeon. This is the idea here, right? So here's a boundary in the ocean that you can see. This is basically ideas that if you drove your sensors across this, you'd be in a really different ocean. And these are the types of boundaries that these are seeing. We're just naming them A, B, C, D, E, and F in this case. We are putting biotelemetry sensors in here. We're basically going to match these seascapes to receiver stations, and we put out 260 tagged sturgeon. So here's the idea, is that Matt was going to put tags in the sturgeon. They were going to swim around these receiver stations, and then he was going to determine whether or not these seascapes or dynamic provinces actually predicted where they were. And what he found is that we could see how much of each one of these seascapes was available. So A was not available very much, so it was hard to use. D was really available, C was really available, E was really available, and then the hash lines here are the actual usage of the Atlantic sturgeon. So if the Atlantic sturgeon didn't care at all about seascapes, they would occur at the same frequency of their availability. In other words, they would just be, you would have this much A would be the same, and that's generally what you have. B and what was available and what they used was about the same. But you can see in seascape C now, these really avoided seascape C. In fact, they preferred seascape E in this case, and that's a prediction. So we could actually map these in real time. We could actually map where these seascapes are, and then with the help from commercial fishermen, we could put these we could put sensors on a robot to go out and actually sense to see whether or not the fish were in the seascapes that we predicted. So you send out a sub that's communicating with us, and it detects a fish. In this case, it would be an Atlantic sturgeon. It sends that information back up to us on shore, and we can now map where these Atlantic sturgeon are according to seascape, these dynamic seascapes. So here are these dynamic seascapes, or these dynamic provinces. I'm going to show you a video that shows you, this is the path of the glider. It's going to move down the coast, and it's going to move down here. It's going to move into this blue area that we think of where the Atlantic sturgeon actually are, and you'll see what happens. So once we get down to these blue areas, we start detecting them. That's what those dots are. These are actual Atlantic sturgeon detections, and we know how much is available versus how much how much habitat is available versus how much we're sampling with the glider, and how frequently we're actually detecting these Atlantic sturgeon. And in fact, what you get is more or less the same result. We show by putting vehicles in the water that in fact these Atlantic sturgeon prefer seascape E, right? Now, we can do the same thing. We can say, okay, what does seascape E actually even look like in these regions? How frequently is it actually found? And like, there's lots of seascape E here along the coast, right along the coast. Seascape C they don't like so much, and we can compare those sort of with. We actually had some detection lines here, and we could see how often we were actually detecting them based on our predictions. And so here's the basic idea, that these Atlantic sturgeon prefer seascape E. What is seascape E? Well, this is the beauty of these dynamic provinces, is that we're not exactly sure everything that goes into them, but we know that they are distinct categories that are being selected for by organisms. And so that's why it might work a little better than temperature or just chlorophyll or just bottom oxygen or something along those lines, is that these conditions together, this temperature and these radiances, are indicators. They're indexes for what the ocean is actually like. And in fact, we can make predictions of this endangered species, say Atlantic sturgeon, fairly easily. We can throw these maps out there and say this is where we think this organism might be. So let's get practical. What do you actually do with this kind of information? I don't know. Well, let me just think about it this way. You go on a cruise and you measure stuff. You go on some epic cruise that starts up here in North America and you sail all the way down to Antarctica. And you want to see whether or not calcification in some phytoplankton or zooplankton species is some function of pH, right? And so you go on a cruise and you get data that looks like this. Calcification versus pH is just sort of a scatter plot. And so your advisor calls you in And he says, how's your research going? This is from PhD Comics. And you say, slowly, I've gotten 3 data points in the last 60 days. Let me see. I think there's a clear trend happening here. There is? Yes, your chances of graduating are asymptomatically approaching 0, right? So you gotta produce, right? So then you think, oh, I remember that lecture about dynamic seascapes and provinces. And I know that I crossed all these different types of excuse me. I know that I crossed all these different types of water as I went through here. And so what I can do then is actually control for my expectations. In other words, because it's a different habitat or biome, I expect things to work a little bit differently. If we go all the way back to our example with the baboons in Delaware, not finding baboons in Delaware is not a big deal because they're not supposed to be here. Maybe calcification rates are supposed to be low here, and they're supposed to be high here, and they're supposed to be a little bit lower over here, and you could use seascapes to do that. And so this same analysis, this is the same points I'm showing you, these are just partitioned by seascape now. In other words, you can actually run a mixed model with random intercepts. And that's what this form is over here. Just calcification is some function of pH, but that is dependent on what kind of seascape or province you're in, and that's what these 4 different lines are. And in fact, you can do the same thing with random slopes. And so it's one way to try and take a look at data that looks like it doesn't have any meaning whatsoever and make some sense out of it, especially if you're able to control for these different environments. And then you're very happy.